DNA, deoxyribonucleic acid. We're going to take a look at what DNA is, how it works, and how it's put together. So one of the first things that we're going to do is take a look at the role of DNA. DNA is the instructions for how your cell works in making proteins. Those instructions are in the form of genes. You have 46 chromosomes, or 23 pairs, containing all of those instructions. Parents pass it off to their offspring, so it's our genetic information. And these genes determine all of your traits, everything from blood type, hair color, skin color, to the types of enzymes or proteins that each cell needs to make. And if you remember back, for eukaryotic organisms, all eukaryotic cells have a nucleus, and this is where the DNA is kept. If you were to look at one cell in a human and pull out its DNA and kind of unwind it, you have about a meter of DNA in each cell. So taking out all of the DNA from every single cell, unwinding it, it's going to be about 9 million kilometers, basically going from the Earth to the Moon and back 13 times. That's quite a bit. So how does it get that way? Well, it winds up. So if we take a look here, we can see that DNA winds. It coils. So it starts a coil here, and then the coil coils, and the coil coils, and the coil coils. So it keeps coiling, and coiling, and coiling, and coiling, and coiling until it can fit into the small chromosome and fit in your cells. Let's look at the DNA structure. Here we have subunits. The subunits are called nucleotides. We have 4 different nucleotides in DNA. We have purines, which are 2 rings, and we have pyrimidine, which are 1 ring. Our purines are 2 ring guys, are adenine and guanine. Our 1 ring guys, or pyrimidines, are cytosine and thymine. There was a guy, Chargaff, who figured out that A and T were in equal proportions and C and G were in equal proportions, and thus kind of came up with this idea that C and G paired together and A and T paired together. So let's summarize that. Our purines, A and G, and our pyrimidines, C and T. A purine is always going to pair with a pyrimidine. So cytosine pairs with guanine, and adenine pairs with thymine. Looking at the structure of DNA, we have a backbone. The backbone is made out of sugar and phosphate. The sugar that we have is called deoxyribose, kind of fitting for the name deoxyribonucleic acid. And then you have a phosphate in between those sugars. So this is a sugar, this is a sugar, this is a sugar, this is a sugar. And then our phosphates are here, in between. This forms the side of the ladder. The rungs of the ladder consist of nitrogen bases that stick out. We have our G, our T, our C, and A. Depending on the order of those letters will depend on what that gene codes for. This is just one side of the ladder. This ladder has 2 sides. So let's take a look at the 2 sides of our ladder. Here we go. 2 strands stuck together. So we have our one side here, and then we have our other side. Here's the other side. And then you have the rungs in the middle here, giving our ladder. Each strand is held together by hydrogen bonds. C has 3 hydrogen bonds between G and itself. A and T have 2 hydrogen bonds. Remember, A pairs with T, C pairs with G. C having 3 hydrogen bonds, A and T having 2 hydrogen bonds. The winding, the winding coil is called a double helix. So the shape of DNA is a double helix. Watson and Crick are known for this model. They are known for kind of coming up with this idea, except for there's a little bit of controversy behind this. There was another scientist working on this at the same time in the same building. Her name was Rosalind Franklin. Watson and Crick essentially stole all of her work, published it before she could, and kind of stole her glory. So let's give some props to Rosalind Franklin as well for doing all of the footwork that Watson and Crick stole. So our two functions of DNA: duplicate, basically copy itself, and remember this happens in S phase of mitosis. And then also to store the genetic information, basically all those instructions on how to make all the proteins in your body. So let's look at that duplicate first. Remember, we have our DNA, has two strands. So it has one strand here, and then it has a complementary strand here. So A, remember, pairs with T, and G pairs with C. They complement each other. So this is important when we're copying our DNA. If we take a look at this one strand of DNA, just basically the order of the letters, we can write the corresponding or complementary strand below it. So A pairs with T, T pairs with A, G pairs with C, C pairs with G, and so on. So we can write the complementary strand below that piece of DNA. And this is basically what happens when we copy our DNA. We are looking for that complementary strand. So where does DNA replication happen? Remember, DNA is kept in the nucleus and it never leaves the nucleus. So it happens in the nucleus. When? Before it divides in S phase of the cell cycle during interphase. Why? Because the two new cells need an exact copy. If they don't have an exact copy, they can't function correctly. And the how. So the how, DNA unzips, so it breaks those hydrogen bonds between the bases. Complementary bases will come in and bind to the template strand, basically that original strand. We're going to have bases come in and join up. DNA polymerase is an enzyme that connects them together. So DNA polymerase, that enzyme, is going to connect those nucleotides, and then the helix will reform. This is a basic of what happens in this next picture. Here we have our strands unwinding. So here is the strand unwinding. This blue circle is our DNA polymerase, that enzyme that's attaching those new bases. So there's bases kind of in the cell, and they're going to attach the complementary strand. So remember, A is complementary to T, and C is complementary to G. So that DNA polymerase is going to come in and read the template strand. If there's a G, it's going to attach it with a C. If there's an A, it's going to attach it with a T, and so on. Our second function of DNA is basically all of the instructions to make the proteins in your cell. These are your genes. And there are lots of genes on every single chromosome. Genes code for proteins. Proteins are made in the cytoplasm. The instructions are in the nucleus. So we somehow need to get a copy of that DNA from the nucleus to the cytoplasm. Since DNA never leaves the nucleus and ribosomes are out in our protein is made out in the cytoplasm on the ribosomes, we need to get a copy there. RNA is our copy. So we're going to make RNA in order to bring all of that information we need to that ribosome to make that protein. RNA is ribonucleic acid. So the sugar here is a little bit different. Instead of deoxyribose, here the sugar is ribose. That is one big difference. We need to have the sugar, something different on these molecules, or else the body is going to think that copy of RNA is actually DNA itself. So we make a few slight changes, one of them being that the sugar is ribose instead of deoxyribose. It's very similar to DNA. The building blocks are very similar. So it has the A, it has a C, and it has a G, but instead of the T, it has U. So here is our differences. We have ribose instead of deoxyribose. It is a single chain, so it's not double. And we have U instead of T. So anytime that you're going to have an A is going to pair with uracil, or U, instead of our T. So if you see U's, you're looking at RNA. If you see T's, you're looking at DNA. We have three main types of RNA. We have mRNA, or messenger. We have rRNA, or ribosomal. And we have tRNA, or transfer. Later, we will talk about the functions of each of these three types of RNA.